To all participants, thank you for standing by. The conference is ready to begin. Good day, ladies and gentlemen, and welcome to the webinar on ICRP's recommended dose limits for the lens of the eye. I'll turn the meeting over to Caroline Purvis. Please go ahead.
Welcome, and thank you for joining us today. My name is Caroline Purvis, and I'm the Director of the Radiation Protection Division at the Canadian Nuclear Safety Commission, otherwise known as the CNSC. I'm delighted to introduce the first webinar organized by the CNSC in partnership with the Canadian Radiation Protection Association. Today, we will hear from 4 speakers that will provide expert opinions and perspectives regarding the scientific basis for the dose limits for the lens of the eye, as recommended by the International Commission on Radiological Protection, or the ICRP. Before we begin, I'd like to take this opportunity to thank the speakers for taking time out of their busy schedules and volunteering to participate in this important day. The format of the webinar will include presentations from each of our guest speakers, followed at the end by a session of approximately 30 minutes where we will do our best to respond to questions. We will try to address as many questions as possible. If you wish to ask a question, please send it by email at any time during the presentations to the following web email address. It's up on your screen currently, but just to confirm, the address is cnsc.webinar.ccSN@canada.ca. As I said, for your reference, the email address is provided on the slide, which currently appears on your screen, and it may also be found on the CNSC webpage dedicated for the webinar. With that, I'd like to introduce the first speaker, Dr. Norman Kleiman. Dr. Kleiman is the Director of the Eye, Radiation, and Environmental Research Laboratory at the Mailman School of Public Health within the Department of Environmental Health Sciences at Columbia University. Dr. Kleiman is also the Director of the MS degree program in Radiological Sciences. Dr. Kleiman's primary research interests revolve around the examination of the effects of environmental exposures on eye tissues, including a variety of radiation exposures. Dr. Kleiman has considerable expertise in measuring radiation damage to the lens in both human subjects and experimental animal models. Dr. Kleiman has served on committees of the ICRP and NCRP, which is the National Council on Radiation Protection and Measurements, tasked with providing guidelines for ocular exposure limits, and has published extensively on radiation cataract risk for interventional cardiology medical personnel. Dr. Kleiman's presentation today will summarize the effects of ionizing radiation exposure on the eye, the types and presentation of lens damage, the methods used to assess radiation-induced lens changes, and the generally underappreciated potential visual risks associated with cataract surgery in relatively young individuals still in the workforce. Welcome, Dr. Kleiman. Please proceed when you're ready.
Thank you. I appreciate the opportunity to discuss this topic, and welcome to everyone listening and to my co-panelists. So, just to remind everyone, the purpose of radiation protection is to prevent, as it says here, deterministic events of clinical significance and to limit any stochastic events to a level that we find acceptable within our society and within our occupations. And to define deterministic events, these are effects that occur above a certain dose, and a typical example is reddening of the skin or, if severe enough, a radiation burn. Whereas stochastic events have a probability that increases with dose. At zero exposure, zero risk. At very low exposure, very low risk, with a probability that increases. And typical examples of that are transformation of cells, mutation, and carcinogenesis. So one of the fundamental questions is, where does radiation cataract fit in this scheme? Prior to 2011, exposure guidelines for the eye were based on the fact that radiation cataract was thought to be a very deterministic event with a relatively high threshold dose. And if we look at the history of ICP recommendations for dose limits to the lens, we see in 1977, the annual exposure limit was 300 millisieverts. 10 years ago, that limit was reduced to 150 millisieverts, and the corresponding cataract threshold was also reduced from 15 sieverts to 5 sieverts for an acute exposure, but also noted that a protracted exposure had a higher putative threshold dose. In 2012, the ICRP reevaluated the data, primarily the human epidemiological data, which Dr. Shore will go through in some detail, and reduced the annual occupational exposure limit to 20 millisieverts per year, with a putative cataract threshold of 500 milligray, without any distinguishing between acute, protracted, or chronic doses. And this is the actual statement released by the Commission in 2011, in particular for radiation cataract, noted 20 millisieverts per year, averaged over 5 years, with no single year exceeding 50 millisieverts. The IAEA adopted the ICRP guidelines shortly thereafter. The NCRP in the United States recently reconsidered the evidence from the ICRP and adopted a somewhat higher recommended exposure of 50 millisieverts per year. But throughout the world, there was some disagreement. The IRPA questioned the new guidelines and the strength of the epidemiological evidence, and the EPRI, quoted here, says there's not currently enough available information and would prefer that additional research and additional human epidemiological studies would be done to reevaluate whether or not chronic exposure thresholds were in fact the same as acute exposure or protracted exposure. So it's essential that we have some information about the accurate dose threshold for eye damage so that we can really effectively assess radiation risk to the eye and in particular to the lens. So how did we get these guidelines for lens exposure? And Dr. Shore will go through this in some detail, but just very briefly, over 100 years ago, an investigator named Chalopecki reported cataracts in rabbits who were exposed to X-rays, but curiously decided that this wasn't due to radiation exposure. Shortly thereafter, however, a number of individuals reported radiation cataract in humans and in experimental animals. Some of them are shown here on the lower right. The two large studies, surveys of atomic bomb survivors in Japan starting in 1950 and proceeding for the next to date, actually, and early studies in physicists who are involved in cyclotron research better define the kinds of changes that occurred in the eye after radiation exposure. But these studies were somewhat limited because they had very few subjects with very low doses, relatively short follow-up, and relatively insensitive diagnostic techniques. And over the intervening 50 or 60 years, more recent studies were consistent with much lower threshold models for radiation cataract. And some of these are shown here. I'm sure Dr. Shore will go through these in more detail. This has led over the last decade to a plethora of reports on radiation cataract in occupational settings, just a few shown here. And one of the unique features of the lens is the fact that it's accessible non-invasively, and we can measure changes in the lens over time, allows us to examine environmental and occupational, mechanistic, and genetic influences on radiation cataract in humans and also in experimental animal models. In my work in interventional cardiology, we've done quite a bit of work in assessing exposure risk for interventional medical personnel. And as Dr. Ainsbury will talk after Dr. Shore, there's been quite a bit of interest in study in using experimental animal models to better understand the mechanisms by which radiation cataract forms. So why the lens? Why are we interested in radiation cataract? Well, the lens is one of the most radiosensitive of all the tissues in our body. We've known that for many years. And in fact, it's much more radiosensitive than any other tissue in the eye. And you can see here the dose in gray is for any minimally detectable change in any of these eye tissues, and the minimal dose for actual visual changes in the eye. And you can see the lens is several orders of magnitude more sensitive, for example, than the retina. So what is cataract? Cataract is very simply a change in transparency of the lens. And there are many forms and presentations of cataract and many different etiologies, and this is a beautiful drawing by an ophthalmic illustrator from the 40s showing the great variety and presentation of cataract. And these are all pictures, documented pictures of human clinical cataracts that the artist illustrated in this picture. So cataract is certainly a global issue. It's the largest cause of visual disability worldwide. It's certainly an increasing economic burden as people live healthier, longer lives. In the U.S., it's 12% of our Medicare budget. It's 60% of all Medicare visual costs. Most of us have relatives who, as they age, get cataract. But we're not talking about aging-related cataract. We're talking about a specific subset of lens opacities, which we term radiation cataract. And I would define a radiation cataract as an opacity of the lens that most often originates at the rear of the lens along the visual axis, and we call that a posterior subcapsular cataract. It occurs in the posterior subcapsular region of the lens. And in this picture, you can see a bright opacity here on a slit lamp image. This is the lens shown here. This is the anterior surface of the lens, the posterior surface of the lens. This is a relatively clear lens except for this small discrete opacity at the rear surface, just anterior to the lens membrane here, the lens capsule. This is the corneal surface. This is the anterior aqueous. This is the anterior surface of the lens. This is the posterior surface of the lens. This is a classical picture of a radiation cataract. So why do we care about radiation cataract? We're certainly concerned about its health impact on workers. It is a pathology occupationally which we may be able to prevent or retard by the use of appropriate protective gear. There's some question about whether it tells us something about exposure to the body and certainly exposure to the head in an unprotected worker. And it's an excellent model for low-dose exposures. So I would argue that radiation cataract is an excellent way to study potential human health risks following occupational low-dose ionizing radiation exposures. And certainly there have been many investigators around the world who've taken advantage of this. Another point I'd like to emphasize, however, and it's probably not addressed in many of the publications about radiation cataract risk, is the potential visual disability and the morbidity resulting from the actual surgery to repair, to correct radiation cataract. It's greatly underappreciated, and we'll talk about this at the end of the talk. So let's back up a second. Let's talk a little bit about the lens. This is a typical picture of a primate eye. This is a human eye. The lens occupies a relatively small volume of the entire eye. Primary purpose of the lens is to accommodate or fine focus light onto the fovea, the point of our greatest acuity in the eye. If we look at a cartoon of the lens, we see that the lens can be divided into three regions. The center of the lens, which is called the lens nucleus, the cortex of the lens, which is the outer portion of the lens, and the posterior region, where we have a posterior subcapsular cataract formation. The lens has a single layer of epithelial cells on its anterior surface, and in a discrete region of that epithelium, somewhat toward the equator, the cells divide, and curiously, as they divide, they only move toward the lens equator. And as they do, they differentiate, they grow greatly in size, they orient 90 degrees, and they become a new type of cell called the lens fiber cell. These are living cells, but in their mature form do not contain any nuclei or mitochondria, which is a good thing because it allows the lens to maintain transparency. This is a rather thick structure, several millimeters in thickness, and if it had nuclei and mitochondria in it, light wouldn't penetrate. So it's been designed to serve as a lens, as a fine focus mechanism in the eye, with the only cells that contain DNA in an active form, the anterior epithelium. And only in this region do the cells divide, and as they move toward the equator, they differentiate and become a new type of cell. So there can be opacities in the lens that can form in various regions. Most commonly in aging, one gets a cortical opacity, the outer layers of the lens, the most newly formed lens fiber cells. The risk factor for cortical opacities is primarily aging and perhaps sunlight exposure. A secondary type of cataract that occurs less frequently than cortical cataract is nuclear cataract. That occurs primarily in the center of the lens, sparing the cortical regions in its pure form. And one of the risk factors for nuclear cataract is smoking. Radiation cataract is not associated with nuclear opacities. Radiation cataract is associated with posterior subcapsular opacities, and it's this distinguishing characteristic that allows one to look at what kinds of etiological agents cause posterior subcapsular cataract as compared to nuclear or cortical cataract. Systemic use of steroids is also associated with posterior subcapsular cataract, so that has to be included in any differential diagnosis for a patient presenting with a posterior subcapsular cataract. Some form of diabetes are also associated, small number of that, with posterior subcapsular cataract. But the fact that it occurs primarily in the rear of the lens allows us to distinguish it, for example, from smoking-related causes or from the typical age-related presentation, which starts in the cortical and the anterior regions of the lens. And of course, as cataract progresses with time, and all cataracts tend to progress with time and increase in severity and in the degree of opacification, we can have a mixture of posterior, cortical, and nuclear forms. It's the original initiating form that allows us to learn something about the initiating agent, whether it's sunlight, radiation, or smoking. So posterior subcapsular cataract, we believe, is the cataract that's most commonly associated with radiation exposure. And as Dr. Ainsbury will talk, I'm sure, later, we believe that the pathomechanism of radiation cataract is, in fact, damage to the DNA originating in the anterior region of the lens, and in particular in the dividing cells or the differentiating cells of the lens. We believe this because of a number of animal studies in which the lens or portions of the lens have been shielded during exposures, by studies in which we inhibited mitosis in the lens, and by human clinical examples in which we irradiated the posterior half or the center of the lens, sparing this germinative and differentiating region in which radiation cataract did not form. So hypothetically, we believe that ionizing radiation causes damage to the DNA in the epithelial cells, leading to either aberrant division or aberrant differentiation, leading to abnormal lens fiber cells, ultimately resulting in a opacity, a loss of transparency, which we define as cataract. So how do we measure this kind of damage to the lens? Most commonly, we use a slit lamp, the same device you use when you visit an optometrist or an ophthalmologist's office. You put your head in a chin rest, and the ophthalmologist perhaps dilates your eyes and shines a bright light in them and takes advantage of the specular reflection to visualize changes within the lens. A secondary method is called retroillumination, in which we take advantage of the fact that the back of the eye, the retina, will reflect some of the eye forward, and if there's an opacification of the lens, some of that light does not reflect backwards, so the observer sees a dark spot in the lens where there is an opacity. A more complicated and sensitive method for looking at lens damage is called Scheimpflug imaging. It's a very specialized kind of slit lamp. It's not available in most ophthalmologists or optometrists' office, that allows us to assess changes in density within the lens and gives us very pretty images of lens changes. And finally, starting to be used is a technique called contrast sensitivity. There are different measures of visual function. Acuity, when you go to the ophthalmologist's office and you look at the eye chart under bright light illumination and you read the E or the other letters on the chart, that's a measure of visual acuity under optimal conditions, and it tells us something about our ability to perceive clarity of letters under bright light situations. But for example, in occupational medical settings where an interventional physician is in a dim room looking at a fluoroscopy room, they may have good acuity, but they may have decrements in contrast sensitivity, another measure of visual function. And we believe that radiation cataract selectively affects contrast sensitivity because it affects posterior subcapsular regions of the lens, in contrast to cortical and nuclear cataracts. And there's some evidence that PSCs are uniquely associated with decrements in contrast sensitivity. So So this is a typical slit lamp you might see in an ophthalmologist's office. This is one of my slit lamps. We use this for both human and animal studies. It's a typical slit lamp device. This is a Scheimpflug camera, one type of Scheimpflug camera, which gives us more sensitive densitometric measures of the lens. And this is an example on the right of a typical slit lamp photograph of an interventional cardiologist with a characteristic posterior subcapsular cataract shown here on the right. And the same interventional cardiologist on retroillumination, where we're taking advantage of the reflection from the back of the retina, showing that same opacity, that same spot or discontinuity here seen on reflection back in the slit lamp. How do we quantify? How do we measure these kinds of changes in the lens? There have been a number of methods used over the years. The method I and Dr. Merriam and Wargel historically used is called Merriam-Vogt scoring, which assigns a score between 0 and 4 to the degree of opacity, the location of the opacity, the amount of area of opacity within the lens. We think this is uniquely suited to scoring radiation, cataract, posterior subcapsular changes. More commonly in ophthalmologist's office, LOX3, which is the most recent version of cataract staging, is used. LOX3 was originally designed for nuclear color and nuclear changes. It has some applicability to posterior subcataract, but in my opinion is more uniquely designed for nuclear changes and perhaps age-related changes as well. A variation on quantification was called focal lens defects. This was used in some of the epidemiological work in contaminated buildings in Taiwan. Digital Scheimpflug imaging allows us to get densitometric measurements through the lens. One of the disadvantages of digital Scheimpflug is that it passes a narrow beam of light through a defined region of the lens and doesn't tell us anything about densitometric changes off axis of that beam of light. And finally, contrast sensitivity testing, which I believe is an adjunct to typical slit lamp biomicroscopy. So this is a typical Merriam-Vogt scoring scheme showing changes occurring primarily initially at the posterior region, extending then as the cataract progresses to the anterior region, and then involving a great deal of the lens area by the time we get to a 3+, which is a very serious visual disability of the lens. This is a typical Scheimpflug image of a radiation cataract, allowing us to make densitometric quantification of the changes in the lens from the posterior radiation cataract shown here through the relatively clear regions of the rest of the lens. And lastly, contrast sensitivity testing. The device that we use uses rotationally symmetric targets, and the user essentially pushes a button saying yes or no whether or not they see a target. We can test at a number of cycles per degree under different kinds of luminance. And here's a typical contrast sensitivity image. The subject on the left clearly has a decrement in contrast sensitivity, as shown by the lower dark line, whereas the subject on the right has a perfectly normal contrast sensitivity pattern. So how can we reduce or eliminate radiation exposure risks to the eye, especially under occupational conditions? Very simply, eye protection. Leaded eyewear provides very good protection to the lens of the eye. It is somewhat heavy. An 80-gram lens spectacle is equivalent to 0.75 millimeters of lead, and it's essential, as you can see in this image, that we have lateral protection as well, because a great deal of the scattered reflection is occurring through the side rather than through the front of the lens. And when you combine leaded eyewear with additional shielding, we can get as much as 25% increased protection from scattered radiation to the eye. So certainly here at Columbia in our interventional suites, whether it's radiology or cardiology or urology or neuroradiology, we recommend that the users all wear protective eyewear of some sort. So the treatment for a radiation cataract, or indeed for any kind of cataract, is simply surgical removal. There is no therapeutic intervention. There is no medicine that will slow or reverse cataract formation in humans or in animals. And cataract surgery, by and large, has a very good success rate, which we define as an improvement in vision over that before cataract surgery. It's a very common surgery. It's generally outpatient surgery, and people generally are fairly well satisfied with cataract surgery. However, the visual disability and morbidity resulting from radiation cataract is somewhat underappreciated, especially in a younger working population. For example, an interventional cardiologist who needs good bilateral vision and good contrast sensitivity under low light conditions. So some of the potential surgical or post-surgical complications that can occur after cataract surgery are listed here. There's quite a few of them. What kind of risks do these pose? There are potential post-operative visual complications as well. While you may restore vision to that prior to cataract surgery, oftentimes patients may complain of glare and flare at night, a somewhat reduced acuity compared to what they had many years ago, a decrement in contrast sensitivity, and some issues with bilateral vision as well. Some of the risk estimates in the literature for some of these changes for posterior subcapsular opacification is secondary opacification, which occurs months to a year or two after surgery, is as high as 10%. Treated usually by laser therapy to ablate or remove the posterior subcapsular opacification. Relatively successful in the treatment, but more concerning is cystoid macular edema with a rate somewhere between 1 and 10%, the risk of retinal detachment about 0.5% overall, but considerably higher in younger myopic males, for example, the kind of individual who might be working in an occupational setting. And permanent vision loss. So for someone who requires good vision to see under low light conditions, these may be concerns worth considering when we consider the use of eye protection, leaded eyewear, or other shielding to reduce exposure risk to the eye and reduce the likelihood of any kind of decrement in vision or untoward consequence of cataract surgery. And in fact, the Health Physics Society a few years ago concluded, and I'll quote, the combined morbidity and mortality risks of surgical correction of radiation-induced cataracts, which they estimated to be 1% or more, and the unquantified risk of a physician misdiagnosing or mistreating a patient because of loss of visual acuity due to the presence of undiagnosed cataract greatly outweighs the risk of cancer in affected individuals. So in essence, one needs to take into account the potential post-surgical consequences of cataract surgery and the effect of a small, perhaps not yet requiring surgery, opacity on visual function, and in particular contrast sensitivity in these workers. I think that we need continued follow-up of various occupationally exposed human cohorts combined with continued experimental animal studies to understand the mechanism and better understand what the actual cataract threshold is and how we place radiation cataract in the scheme of deterministic and stochastic events and better inform our ocular risk guidelines. And I look forward to hearing what Dr. Shore and Dr. Ainsbury have as well. I want to thank my collaborators throughout the world, my funding sources, and I always close by recognizing my mentor and my friend Basil Wargel, who passed away way too early. Thank you.
Thank you, Dr. Kleiman, for your very interesting and informative presentation. I think we all learned a lot. With that, I'd like to introduce the second speaker today, Dr. Roy Shore. Dr. Shore is Professor Emeritus at the New York University School of Medicine and retired Chief of Research at the Radiation Effects Research Foundation in Hiroshima, Japan. Dr. Shore served on ICRP's Committee 1, dealing with radiation effects, for 16 years. He has also served on various committees and task groups for UNSCEAR, the United Nations Scientific Committee on the Effects of Atomic Radiation, the World Health Organization, the U.S. National Council on Radiation Protection and Measurements, and the U.S. National Academy of Sciences. Dr. Shore was an investigator on the radiation cataract studies of Japanese atomic bomb survivors and the Chernobyl cleanup workers. Today, Dr. Shore will provide a summary of the epidemiologic information on the dose rate effectiveness factor for radiation cataracts, based on several new studies since the ICRP issued its new recommended dose limits for the lens of the eye. Please go ahead, Dr. Shore, when you're ready.
I would like to talk today about the epidemiologic studies of radiation cataract risk. and in view of the time involved, I won't be able to cover very much in detail, but I hope I can give you a quick overview of the status of the epidemiology. Here's a quick summary of what I'm going to cover in the overview. I'll talk about a few of the main studies, the larger studies that have had an impact on radiation risk assessment. And I'll be concentrating on studies that have dose response data because those are more informative than ones that don't have such information. So I'll look at a summary of the risk estimates and also those threshold estimates. We'll talk a bit about minor opacities versus more significant cataracts, and I'll say a few words about the interventional cardiology workers and raise some questions at the end. So first of all, selected epidemiologic studies of radiation cataract. One principal study is that of the adult health study in the atomic bomb survivors. About 17,000 atomic bomb survivors have been brought in every 2 years for clinical examinations, and those have included examinations for visual defects. And here's one example of a study done in about 2004, looking in this case at the results with regard to posterior subcapsular opacities. And I'm just going to call them PSCs so my tongue doesn't get twisted over posterior subcapsular. But as you can see, there was a dose response for posterior subcapsular or PSC opacities. However, many of these occurred in those who had higher doses, and of course the whole study is about acute doses, acute exposures, whereas many of our interests today are having to do with chronic exposures at low dose rates. As you can see, they were screened at about 55 years after exposure, which means that their average age was 65 at this time. So we're not looking at a young population. We're not looking at those within a decade or 2 decades after exposure. In addition to seeing an excess of PSCs, they also saw an excess of posterior cortical cataracts with a slightly lower risk ratio than for PSCs. In this analysis, they couldn't reject a linear dose response, but the test for that is not very statistically powerful, so it's uncertain how much that means. The other main study from the adult health study pertains to cataract surgery incidence. This was over the time range of 1986 to 2005, which was 40 to 60 years after exposure. The mean age at surgery was 74 years, with a range of 48 to 94. And again, they did not see a significant upward curvature, though there was a slight indication of some upward curvature. Incidentally, the mean dose to the subjects in the adult health study was about half a gray, so it's not a low dose study particularly. Well, let me just briefly mention some strengths and limitations of the atomic bomb studies. For the opacity screening study, clearly they had standard scoring. They took slit lamp photos so that afterwards they could do a systematic review of those photos, which was published in the Nakashima publication. On the other hand, they saw very few high-grade opacities. For instance, only about 4% of the PSCs were what they would consider potentially vision impairing, which meant a LOX2 score of 3 or more. The cataract surgery study was looking at more important, more significant visually impairing cataracts at good statistical power, but because of the fact that when one looks at cataract surgery, there are a variety of reasons that people do or don't get cataract surgery, so the sensitivity and specificity of that assessment as a surrogate for vision impairing cataracts is not perfect by any means. And there was no information on cataract location in that study. And as I already mentioned, for both studies, there was only a high dose rate, even though the doses were fairly accurate compared to some other studies, but it doesn't inform us directly about what happens in, say, occupational settings. Okay, next study I want to talk briefly about is opacities in Chernobyl cleanup workers. The study made a valiant effort to try to assess doses, but there were just a number of things that prevented an excellent dose assessment. They had official gamma dose estimates on all these workers, but only 14% of those were based on actual measurements. So they tried to calibrate the official dose estimates against electron paramagnetic resonance of teeth. They had teeth from about 100 of the workers, and they used that regression between the two to then estimate the gamma doses to the eye for all the workers. But there were additional complications in that they didn't have many measurements at all of beta doses, and there were some work sites where beta doses were fairly prominent. So they calculated estimated ratios of beta to add in. Well, all of this led to substantial dose uncertainties. Well, the results were as shown here for the Chernobyl cleanup workers. First of all, you can notice on the right that for nuclear cataracts, there was no association with radiation, which most other studies have also shown. So we generally do not think about nuclear cataracts when we think of ionizing radiation. They did, however, find excesses for posterior subcapsular and cortical cataracts, but those were primarily at stage 1 using the Merriam-Vogt scale, which Norman already described. If we limit it to those that are somewhat higher on the scale in stages 2 to 5, for all non-nuclear cataracts, you can see it looks like it may be elevated, but it's not statistically significant. The question then becomes, what do we make of all of it if most of them are just the pretty minor opacities? And in fact, there are only a little over 100 people who had opacities, non-nuclear opacities greater than the grade 1. Well, to look a little more closely at what the dose distribution of the cataracts look like, you can see in this example that for PSCs, stage 1, it looks like there were excesses primarily in those with 800 milligray or more. For cortical, maybe 600 or more, and it was not significant. It would just jumped around for nuclear cataracts, or all non-nuclear cataracts, rather, excuse me, with stages 2 to 5. The problem here is that there were only about 380 workers who had half a gray or more of exposure, so the numbers are very small at the higher doses. Well, again, a brief critique of the study, mostly low dose rates. That was a strength, a large cohort, and they did have information on a number of cataract risk factors, as the atomic bomb studies also did. But there were large dose uncertainties, and there were relatively few higher grade opacities. Well, a third study I want to briefly mention is the MIAC nuclear workers. The first publication on this came out 2 years ago, and that publication was only on senile cataract. It was called All Lumped Together. And one can see that the dose response for senile cataract looks pretty good, pretty linear, including perhaps at doses under half a gray. But because it's a mixture of a variety of cataract types, and also because there's a potential for some problems with the equivalence of assessments at different doses, because they were measured they were examined every year by an ophthalmologist as long as they worked. But of course, those who worked longer had a greater potential for higher doses, and so they had more examinations, and so that could cause some confounding in the assessment here. But I think it's going to be an important study. A new paper is currently under review, which will look at PSCs and cortical and nuclear cataract separately. And I think we'll all look forward to that and see what they find once they have a more refined estimation of types of cataract. Well, let me summarize some of the dose response risk estimates. I shouldn't say some of them, all of them that I could find, actually. And I broke it down according to the type of cataract that was being looked at. So on this screen, you can see that those that are in red are for PSCs. And if you look at the relative risks at 1 gray, which is over on the right, you can see that most of the estimates are around 1.5 relative risk as central estimates, other than the China industrial radiographers. If you look down at the bottom, you see the same thing for cortical opacities. And again, the industrial radiographers have a lower risk estimate than the others do. But their estimates are around 1.4 roughly with that study. The China industrial radiographer study was based on quite small numbers. There were only about 30 PSCs and 40 cortical opacities among the radiographers. So that has some limited information in it. If you look at other studies where it wasn't as well defined as to what cataract types were being examined, you find more variation in the risk estimates. And this slide provides that information. You can see in the red values that these are ones where they were doing prevalence screening for cataract. And again, you see the MIAC workers with a relative risk of 1 gray of 1.3, and others range from slightly over 1 to threefold. But some of them were statistically significant elevations and some were not. I don't have time to go into the details of these various studies, but there were real variations in terms of the qualities of the studies. Down at the bottom, those in blue are cataract surgery, and for the U.S. radiation technologists, it was based on self-reports of cataract surgery for some tens of thousands of radiation technologists, and in fact, they found about, I think, between 600 and 700 cases of cataract surgery in that way. Because they were radiation technologists, they probably had reasonably accurate reporting of that. For the A-bomb study, that was a study you already saw, and you can see the relative risk there. Well, a key thing is to look at the dose response thresholds, because that has been a driver of where we set the dose limits. So let's review that briefly. And again, I've broken it down into type of cataract that's being looked at here. The first two in green are the PSCs, and in the A-bomb study, the central estimate of the dose threshold was about 0.7 gray. In Chernobyl, for grade 1 prevalence, it was about 0.4 gray. For cortical cataracts in blue, you can see estimates of 0.3 and 0.6 gray. And then for a mixed, undefined, if you will, types of cataracts, you can see also that it ranges from 0.5 to 1.8 as estimates of the dose threshold. And finally, if you want to think about more significant cataract lesions, the two bottom lines in red, one is the PSCs, where in the screening study, it's for those with a LOCS 2 score of 2 or greater, in other words, eliminating approximately the 75% that were LOCS 1. And you can see that the threshold dose there is about 0.3, and for cataract surgery incidence, it's 0.5 gray. Well, one of the things you notice in all of this is that a number of the different studies reported the lower limit of the threshold as being less than 0. And so one might be tempted to infer, well, then that means there's no dose threshold and there's really simply a linear association, which would not be compatible, particularly with an indication of a deterministic effect. However, if you look at the upper limits on those studies, you'll find that they are very high. And in essence, we're looking at studies that don't have very good statistical precision, and we have a wide range of possibilities. So I think one needs to take those with a grain of salt as to how important the less than 0 indications are. Okay, the last two, maybe I should back up. The last two lines on this slide are showing somewhat more significant cataracts and what we found with those thresholds there. Let's try to look at the broader range of studies that have looked at more significant cataract grade, which I defined as grades 2 or greater or cataract surgery. Unfortunately, one has a very limited ability to look at exactly what you want to in studies because you have to rely on what they reported. So I used a broad category of grades 2 or greater. And first of all, the first 4, which have the numbers in red, are studies where there was a low dose rate and chronic exposures. And what you see there is that all of them have a relative risk slightly above 1, but in no case was the lower confidence limit greater than 1. So it's pretty ambiguous as to whether there's a significant effect for these more significant cataracts. The next three studies, the ABOM cataract study and the two studies of cancer survivors, had high dose rates and high doses, and there you do find significant effects. And finally, I included three studies that really have inadequate dose information, but I thought they were of interest to look at. One is the U.S. radiologic technicians, those who had experience with nuclear medicine, and there was no indication of an excess risk if they had done work with nuclear medicine settings. And if one looked at the frequency with which they did it, there was no dose response either. So there was no indication with nuclear medicine workers. Treatment with radioactive iodine for thyroid cancer, and again, you don't see significant increases in cataract surgery frequency. And finally, a study of CT examinations in Taiwan, looking at then in their registry of how many had cataract surgery or multiple cataract prescriptions. I'm not sure what that is. And they reported relatively high indications of risk in relation to CT examinations of the head. However, it seems pretty implausible. Those who had one or two CTs have a 60% excess, a relative risk of 1.6. That doesn't seem very likely, so I suspect there may be some possible biases in the selection of an appropriate control group or other factors in that study. Well, one question we can ask is, does age and exposure modify the radiation risk of cataract? And the study with probably the broadest range of ages and exposure is the atomic bomb survivors. And here you can see in the two studies that I already showed you dose response associations for, you can see the risk estimates for at different age ranges or different ages. And when they looked at PSC prevalence data, they found no excess at all for those who were over the age of 20 at exposure. However, there may be some problems in this with the fact that they were being examined at different ages in the different age and exposure ranges, because the examinations were all done at around 2003 or 2004, and they were at different ages of exposure. For cataract surgery incidence, again, there's a decrease with age and exposure, and one, I think, would want to have more information about that to have a better quantification of it. Well, let me just mention the studies of interventional cardiology workers. I think Dr. Kleiman mentioned those, and he's been a part of several of those. But the studies came about, I think, because of anecdotal observations that interventional cardiologists had an excess of cataracts, and so they began to examine them. But it's almost impossible to do the study you'd want to do, and so they got volunteers either at professional conferences or, in the case of France, by querying different cardiology groups to ask who would be willing to participate. So it was not a random sample at all, but it was those who volunteered. And they had to reconstruct the doses retrospectively based on self-reports of things like numbers of interventional procedures done, whether they wore protective glasses or used protective shields, percent of time that they wore dosimeters or didn't, or whether they wore them under leaded aprons and so on. So there's a good bit of uncertainty in the doses, and the resulting numbers they could examine were relatively small. Well, I did a very crude calculation based on what was reported as the mean doses and the numbers examined and the numbers of controls, and I then calculated an estimated relative risk at 1 gray as shown based on that self-reported information. And you can see that they all look quite elevated. There are only 1 or 2 that don't show significant elevations. I think it's interesting, however, that in France, which is arguably the best study because they were able to use a sampling frame to actually get the cardiologists in that study, that under the old ICRP dose limits, the 1995 limits, only 1 out of the 106 cardiologists they examined had a cumulative estimated dose that exceeded the 5 sievert limit. But when they looked at the new ICRP recommendation of 1/2 sievert limit, they found that 60% of the cardiologists had already exceeded that limit. So it's clear that there's a problem there, and that something needed to be done. And so I think we have moved beneficially in the right direction in trying to be more aware and more careful about the doses to the lens of the eye received by some occupational groups. Well, we have a lot of unresolved questions. How large is the dose threshold? I reviewed the information I could find in the literature, and it looks like there's a fair amount of variability in that assessment. But I guess I would be inclined to say, well, probably the dose threshold is somewhere between a few hundred milligray and 1 gray, based on the numbers that I showed. But I think there's a lot of uncertainty in that statement. We can ask, are the risks and the dose thresholds the same for acute exposures and cumulative small exposures at low dose rates? And of course, there was a change in the ICRP recommendations from the prior ones to the 2012 ones, where in the 2012 assessment, there was the dose limits were the same for acute and chronic exposures. And the data that I showed had pretty substantial uncertainties and variability, so that I don't think we have, at this point, a clear idea of whether the risks are the same for the acute and low-dose exposures. We clearly need more data. And then the third question is, if we base our recommendations on minor lens opacities, do we come up with the same sorts of risk estimates and the dose threshold limits as we would for clinically significant cataracts? And there's very little information on what we would call truly clinically significant cataracts in the data that are available. So it will be very valuable for further analyses and careful analyses to be done of both existing data and new data that come down the road. Fortunately, as I already mentioned, the MIAC worker study has a new paper under review, and so within, I suppose, the next some number of months, we will see those results. And I am led to believe that the U.S. Radiologic Technicians study is also updating their information and will use improved dose estimates. So that may also provide useful data for us in the reasonably near future. So with that, I I would just like to say that if you later look at this presentation online and have an interest in the references, here they are. And with that, I'd like to thank you very much.
Thank you, Dr. Shore, for your excellent overview of the relevant epidemiology. Before moving on to the next speaker, I'd like to remind the audience to send your questions, if you have any, to the email address that's on your screen, cnsc.webinar-webinar.ccsn@canada.ca. Our third speaker today is Dr. Liz Ainsbury. Dr. Ainsbury is a Principal Radiation Protection Scientist from the United Kingdom's Public Health England Centre for Radiation, Chemical and Environmental Hazards. Dr. Ainsbury has a number of publications on the basis for and operational aspects of the new European Basic Safety Standards, which implement the ICRP's recommendations in full. And she's currently leading an EU-funded project looking at how low doses of ionizing radiation might contribute to cataract development. Today, Dr. Ainsbury will discuss the mechanisms involved in the formation of low-dose radiation cataracts, the impacts of dose and dose rate, and the interplay among radiation, genetic background, and age-related cataracts. Please go ahead, Dr. Ainsbury, whenever you're ready.
Okay, that's great. Well, thank you very much for the introduction and also for the invitation to speak on this webinar. I'm delighted to be involved, and I've been listening with a great deal of interest to the previous presentations. They've been absolutely wonderful, and now I want to focus on something a little bit different, and that is, as you've already introduced, what we know about how ionizing radiation, and particularly ionizing radiation at low doses, might cause or contribute to the formation of cataracts. So one of the advantages of not being first is that a lot of the introductory material will already have been covered, but nevertheless I want to just take a little bit of a step back and introduce some of the relevant concepts for my part of the webinar. And first of all is the concept of the human lens. So as you already know, the lens is a relatively small tissue, but it's hugely important. So the lens is a transparent structure, it's part of the eye, and along with the cornea, it helps to refract light to be focused onto the retina, just so that we can see. The bulk of the lens is made up of fibre cells that, as Dr. Kleiman has already said, have no nuclei or organelles, and new fibre cells, interestingly, are created by the lens epithelium all the way through the life of the eye of the lens, and they're created in the germinative zones, as you can see on the slide here, on the anterior of the lens epithelium. And they're controlled by various growth factors, and that means that the very precise architecture that's required for the transparency is in place. So it's very tight temporal and spatial organization of the lens cells, which means that the lens is transparent. And it's exactly the deregulation of this which leads to formation of cataracts. So again, as Dr. Kleiman has already highlighted, cataract treatment is relatively simple. It's a routine operation in many parts of the world. Surgical removal is performed as an outpatient procedure indeed. There can be complications, including, as Dr. Kleiman mentioned, retinal detachment, edema, certain visual effects such as glare. But nevertheless, normally things go well and you can go back to your normal life. However, despite that, I always like to mention the fact that there are many parts of the world in which individuals do not have access to cataract surgery. So cataract remains the most frequent cause of blindness worldwide. It's important to remember that. It's also very well known that cataract is an aging disease. So most cataracts that you'd see in clinical practice, I suspect, Dr. Kleiman, are caused by changes in the proteins that happen during aging, during normal aging, so increased rigidity and reduction in the ability of the lens to deal with oxidation, for example. So age is a big factor. Also, there's a well-known genetic component, so around 50% of congenital cataracts, for example, have a genetic cause, so something like metabolic diseases and chromosomal or genetic syndromes. Cataracts can also be caused by UV exposure in sunlight, drinking too much, smoking too much, all the fun stuff, persistent use of certain drugs, corticosteroids, for example, and, as we already know, ionizing radiation, and that's what I'm going to focus on from here on in. Again, this is a little bit of repetition, but this is my take on it. And the figure I have here on the slide in the top left corner is certainly not as lovely as the figure that Dr. Kleiman shared with us about the types of cataract. As we already have heard, ionising radiation is generally, but I'd like to say not exclusively from the epidemiological literature, associated with posterior subcapsular opacities. There are one or two studies which link cortical opacities with ionising radiation exposure as well. That remains to be seen. But we can, of course, classify cataracts into these three rough terms. So the cortical cataracts have these spokes that come in from the cortices, the edges of the lens. Nuclear cataracts are exactly that, at the nucleus, at the centre of the lens. And the PSC, you already know very well what that means, posterior subcapsular cataracts, on the capsule at the back of the lens. And it's an incredibly well-established paradigm up until very, very recently that radiation cataract has been thought of as a deterministic late effect of ionising radiation exposure. The ICRP, and even in 2007, summarised the threshold for radiation-induced cataract as being on the order of around 2 Gray for acute exposures or around 4 Gray or higher for fractionated or chronic exposures. And anybody with a radiation biology background will know Eric Hall's book very well, Radiobiology for the Radiologist. I always think of this as the sort of Bible for radiologists in some ways. This says in there, radiation is a deterministic late effect, so generations of radiation biologists have grown up with this understanding. However, as you know, recent events have suggested that this may not be the case. Indeed, from a mechanistic point of view, we're starting to see evidence that the radiation cataracts may be a stochastic effect. So certainly there's a lot more to this story than we thought there might be. So I'm going to focus chiefly in this presentation on how we think, or what we know about, how ionising radiation might cause cataracts. Now, the target cells, as Dr. Kleiman said, we believe are the cells in the germinative zone on the anterior of the lens at the edges of the epithelium. And I put a question mark there just because in other tissues, we're starting to get a bit more information about non-targeted effects of ionising radiation. But at the moment, there's really very little evidence at all either way in the lens. So certainly the most likely and most well-accepted paradigm is that it is the germinative zone, so that's what we sort of focus on for our hypothesis generation. Potential mechanisms, actually, if you look through the literature, there are rather a large number. I don't expect you can see the text very well in the figure on the right-hand side of the slide, but I'll give you the reference at the end to look at these things in more detail. But you can summarise these things into rather a large list of potential mechanisms that might be involved in radiation cataract initiation or development. And I'll talk a little bit about some of these, certainly not all of them because we don't have a great deal of time. But it's also important to note that when we're talking about radiation cataracts, there are a lot of things about the radiation that are important as well. So dose, certainly, dose rate, likely to be, and all of these other influencing factors, particularly age at exposure and genetic background. Okay. So the first thing that I'd like to highlight is oxidation, and this is probably one of the most well understood parts of the likely mechanistic chain. So reactive oxygen species appear as a result of ionising radiation in most tissues, if not all, and several other factors, including, but not listed on the figure, resulting in diminishment of antioxidants, so as a result of ageing, for example. And we know that reactive oxygen species cause degradation, cross-linking of proteins, aggregation of these lens proteins, and also DNA damage in their own right, which we hypothesize could lead to this aberrant lens epithelial cell division, cell migration, differentiation, and cataracts ultimately. The figure gives quite a lot of detail about redox regulation in the lens. I won't go through the listed factors in detail, but these factors work together to try and detoxify and control intracellular levels of reactive oxygen species. And the reference given here, Hamada and colleagues 2014, gives a very nice summary in a lot more detail if you are interested in the detail of that. And next, again, Dr. Kleiman highlighted that we are fairly convinced certainly from other tissues, but in the lens itself, that DNA damage and repair mechanisms are likely to be involved. So certainly there's a huge amount of evidence in the lens at high doses, but I wanted to highlight this study by Markovits and colleagues in 2015 that really for the first time demonstrated low dose responses for DNA damage in the lens. So I don't know how well, again, you can see the figures here, apologies for that, but we have doses down to sort of 20 milligray on the figure on the left-hand side and 140 milligray up to around 2.3 gray on the right-hand side, looking at DNA damage markers. These markers, gamma H2AX, 53BP1, allow us to visualize the sites of DNA double-strand breaks, which are characteristic of ionizing radiation exposure. And what Markovits and colleagues found, not only this low dose response, but also that the lens, certainly the peripheral regions of the lens, where we have the germinative zones, where the new lens fiber cells are being created, is more sensitive than the central zone of the lens, as you might expect, but also more sensitive to formation of these double-strand breaks than circulating lymphocytes. And I haven't got it on the slide, but in lymphocytes, these DNA damage, these double-strand breaks are repaired more quickly than they are in the lens. So that's very interesting. And this was done in human lens epithelial cell lines and also in mouse models. So it's quite interesting. Next, I want to say something about intracellular signaling. So tumour-related factors, but also DNA repair factors, immune response factors, and intercellular interactions have been highlighted in this review of 2015 to potentially be involved in cataractogenesis. So it's known that in contrast to most tissues, the lens continues to grow throughout life without ever developing tumours. So to my knowledge, there has never been a cancer of the lens. And from the literature, there's now mounting evidence that mutations of oncogenes, these tumour suppressor genes, the DNA repair genes, etc., may affect cataract development. Association of these factors with cancer have long been recognised, and this really highlights that cataractogenesis may well share some common mechanisms with carcinogenesis. The figure gives the possible changes in carcinogenesis mechanism-related molecules and other events that facilitate spontaneous cataractogenesis. And just to say that the orange highlighted areas, these things here, for example, represent increased gene expression, for example, or increased frequencies of events like cataractogenesis, and the blue highlighted areas represent decreases, so decreased gene expression, for example, or frequency of events. And again, there's a huge amount more detail in this very, very nice review from 2015 if you are interested in that. Then the next thing I just wanted to highlight was stimulation of proliferation. The reason for this is that there are a few studies now who have considered this sort of thing. So in Fujimichi and Hamada's 2015 paper looking at various effects in lens epithelial cells compared to other cell lines, they found something rather interesting and unexpected, that radiation not only inactivated clonogenic potential, but it also stimulated proliferation of these surviving unactivated cells. So these cells are not supposed to go through proliferation, they're supposed to be quiescent. This is a very abnormal observation and not something that we'd expected to happen. So if you look back to the historical data, irradiation, we know very well that it induces excessive proliferation of rabbit lenses at high doses, for example, and we know also that suppression of lens epithelial cell divisions inhibits cataractogenesis in other animal models, so frogs, there's a lot of famous studies done there. If we look again at some work by Markovits, interestingly we have, if you look at the cell densities, again apologies, the size of the text here is rather small, but you can zoom in at a later time. The size of the cell density, the markers EDU and cyclin D1, which are markers of proliferation, we have this rather unexpected increase in proliferation in both central and peripheral regions of the lens at around 250 milligray, and then a sort of reduction in proliferation at the higher and lower doses. So that's, again, something that's quite unexpected, but it's clear that irradiation is having an effect on proliferation, which is something over and above the sort of standard paradigm of a linear response to radiation. Okay. Then I want to highlight some publications on lens protein modifications. abnormal lens protein accumulation is what we think leads to aggregation, and then the lens scatters light instead of focusing on the retina and affects the vision adversely. Quite a lot is known about the formation, structure and function of the lens protein. So the crystallins have a role in transparency, the connexins required for metabolic homeostasis, for example, the aquaporin water channels and cytoskeletal structural proteins as well. And these are hugely important. It's worth noting that protein changes that occur in irradiated lenses are very similar to those that are seen in old age. So as we age, as we already know, the lens gets less good at doing what it's supposed to do and cataracts occur. And what we can do by irradiating the lens, it's hypothesized, is sort of speed up this aging process almost. Also, it's been observed recently that abnormal cellular proliferation that we've already talked about has a role. So if we focus on what's happening in protein modifications, the tumour suppressor gene CRPA, which is associated with protein P53, and that's involved in regulation of cell cycling, DNA repair and apoptosis, and this protects against posterior subcapsular cataracts. That's something interesting going there. Then lens crystallins undergo various post-translational modifications, so all sorts of things that I won't talk about in detail there, many of which also occur in the aging process. So this points to the sort of idea that perhaps what ionizing radiation is doing is speeding up that aging process. Then finally, I just want to focus very briefly on some wider genetic data. So there have been actually a relatively large number of genetic studies in the area in recent years, perhaps because animal models which allow us to do these types of studies are readily available for this work. Heterozygosity of the ATM gene, for example, occurs in something like 0.5 to 1% of the Western population. The ATM, RAD9 and BRCA1 genes are known to be critical to pathways controlling things like DNA damage response, signalling, repair, apoptosis as well. And there's certainly evidence that those with heterozygous mutations of these genes are at increased risk of certain health effects, so they have elevated tissue responses to ionizing radiation and are more susceptible to cancers. The studies that I've highlighted here, amongst others, show that double-strand break repair deficient ATM-mutated mice demonstrate earlier and increased incidence of cataract following ionizing radiation exposure compared to normal wild-type mice. And again, I won't talk in any great detail about each of the studies, but if you look at all of this data together, they indicate that the stress response, the DNA repair pathways and radiation sensitivity are very dependent on genotype. So this gives a strong support to the genetic component contributing to cataract development, and indeed perhaps could be interpreted in support of a stochastic mechanism contributing to cataract development. So a little bit controversial, but that's one way to interpret this data. Okay, so that has been an incredibly fast sort of whistle-stop tour of some of the current mechanistic data that we have, and putting that all together allows us to summarize a rather incomplete and basic hypothesis. So you might say that ionizing radiation, well, we know, induces reactive oxygen species. This and the ionizing radiation itself can directly cause damage to the cell, to these dividing cells that are forming the new lens fiber cells from the epithelium. There might also be protein, lipid or extracellular matrix, ECM damage. There might be wider systemic effects as well, something we know almost nothing about for the lens. And certainly there'll be a genetic component which was controlling how the lens reacts to all of these things. But ultimately, somehow, all of these things, the ionizing radiation is leading to abnormal differentiation and migration, perhaps because the antioxidant capacity might be overwhelmed within the lens, which occurs certainly during aging. And this leads somehow to radiation cataract, perhaps with some post-translational effects as well. So a lot of questions, but certainly we do have a working hypothesis for what might be going on. Then, as I said, I've just covered a few of the potential areas that have been looked at in the literature. There's actually a huge amount of information out there if you put it together. We do have a very good understanding from the wider literature of the lens biology, the structure, physiology, and how lens protein fibers are formed throughout life. We know from the epidemiological studies, as has already been discussed, that radiation causes posterior subcapsular cataracts and opacities are above all others. And we actually know from the mechanistic data a lot about the high dose responses. So we know, I haven't talked about it really, about the impact of relative biological effects, linear energy transfer and dose rate. We know that those things are important. But for low dose ionizing radiation cataracts, there are a number of potential competing parallel mechanisms that we can take from the wider cataract studies. And again, these are summarized in the figure that you won't be able to read, but I will give you the reference for. We know from cellular and tissue level studies that genomic damage is highly likely to be involved. There's certainly an element of genetics in play, and the age dependence is hugely important. And then again, as Dr. Kleiman has highlighted, we know quite a lot about how to detect and assess and classify cataracts. So that's not looking too bad. However, if we then go to the slide which summarises what we don't know, I think this is a rather longer list actually. So this summarises the questions that we think can be addressed experimentally at different levels of research model. So at the in vitro level, so animal and human cell lines, for example, we think we can use these to try and answer questions around whether radiation responses might be stochastic or deterministic. So these are radiation responses rather than necessarily cataract induction, but certainly plugging in some of these gaps, filling in the jigsaw a little bit as to what ionizing radiation is doing to lens cells. We can certainly use animal models not only for genetics but for aging studies, for further development of opacity monitoring techniques, and then we absolutely also need the human studies, the exposed human cohorts, to tell us a little bit about how these other factors have interplay with ionizing radiation exposure, and indeed with the exposed populations, how ionizing radiation may cause cataracts as well. So I just wanted to finish by highlighting our current research project that's funded by the EU CONCERT project, and saying that we are attempting to answer some of these questions.